Hi, everyone. So we're continuing with our Chapter 17 lecture. And again, just a reminder, if you are watching this in the fall of 2020, you do have a quiz on Friday, which is October 30th. Of course, this quiz will occur at the beginning of class during your scheduled class time, and it will cover only Chapter 17. Okay, so when we left off, we were talking a little bit about the fact that we'd gone through the process of transcription in prokaryotes, but we were reviewing the fact that eukaryotes take the basics of transcription that exist in prokaryotic cells, and then they add a few things to it. One of the first differences that I mentioned to you is that in prokaryotes like bacteria, we only had one all-purpose RNA polymerase. But in eukaryotes, there are 3 different RNA polymerase enzymes, each transcribing a different type of RNA that we'll be talking about in this half of the lecture. So RNA polymerase 1 transcribes ribosomal RNA. RNA polymerase 2 transcribes messenger RNA, and RNA polymerase 3 transcribes transfer RNA. Of these 3, only messenger RNA is going to be translated into protein, okay? The second difference in transcription that we're going to talk about in eukaryotes has to do with the fact that the structure of eukaryotic genes is much, much different than that of prokaryotic genes. So in our previous lecture, I had drawn you a little diagram, but for review purposes, let's look at this better diagram than the one that I drew you of the structure of a typical eukaryotic gene, okay? So the first thing I want you to notice about this eukaryotic gene is that it's kind of like we've taken the idea of a prokaryotic gene and just added onto it, okay? So I'm going to say that this is all the gene, right? And just like in a prokaryotic gene, we have a promoter area and we have kind of a transcriptional unit or a coding area here. But you can see that all of this is super expanded in eukaryotes. So for example, this whole area here, I'm going to call the promoter, even though your diagram limits the promoter to just this region here. But essentially all of this region upstream here, all of this ginormous region is involved with recognizing the beginning of a gene. So let me just change colors here so you don't get confused. All of this region here is involved with things that are going to attach to recognize the start of a gene and get transcription going. Once transcription gets started, then you have this transcriptional unit here. Now, I'm not going to call it a coding region. Instead, I'm going to call it a transcriptional unit. And that is because we have some non-coding areas. We have what I called a 5' untranslated region, or UTR. We also have a 3' untranslated region. So in other words, these are regions that are copied via transcription, but they are not translated into protein. Then here in the middle, we have some sequences called introns, which are in pink here, and exons, which they've done in green. Only exons are going to code for proteins. Exons are excellent and introns are insignificant. And then in this diagram, you can see that sometimes you'll even have additional features that go beyond the transcriptional unit. So let's look at the simplified diagram that I drew for you guys. And just to walk you through it one more time, this entire block here is what I am calling a gene. Okay, so I'm just going to kind of go back over it for you. This is your gene. All right. At the front end of our gene, we have a promoter region that is involved with recognizing where the gene starts and getting the process of transcription started. I mentioned for you that at the -30 position, there's a feature called a Tata box. It's kind of similar to the features that we saw in the promoter of bacteria, but there are, as you saw in my earlier diagram, additional sequences upstream from here in this promoter that are involved in recognition of the start of this gene. So promoters in eukaryotes are far more complex. Then I told you, okay, so right here is what we're going to call our transcriptional start site. And then this whole region, I kind of called it the coding region, but let's use your book's terminology. I like that. Let's call it the transcriptional unit. I'm going to make that a little thinner. Let's call it the transcriptional unit. Because not everything in here actually codes for protein. So you'll recall that this entire region here, going all the way down here, is what we talked about in our last lecture. And again, I told you that at the beginning of this transcriptional unit and at the end of this transcriptional unit, we have untranslated regions that are copied via transcription, but they are not translated into protein. So we call this the 5' and the 3' UTR. Then, interspersed in between these two bookends, we have a series of exons here and introns here. And what these are chunks of DNA sequence, but the exons actually code for protein while the introns are non-coding. So this one codes, and this one codes. Finally, at the very end of this transcriptional unit, you'll find a sequence here called the polyadenylation signal. That is a sequence that tells an enzyme to cut an mRNA transcript, and we're going to see that it has an additional function a little bit later. So all of this is to say that while the structure of prokaryotic genes is simple, the structure of eukaryotic genes is complex. So the next difference in transcription is that we're going to have to do some mRNA processing to make a functional messenger RNA molecule. Hopefully you can see that we're going to have to do some editing. If we're copying from here all the way down to here, right here, but not everything is getting turned into protein, we're probably going to need to edit out some of this sequence. And that is exactly what we find. All right, so let's back up. Let's go back to this idea that promoters are larger and more complex in eukaryotes. So there is no sigma factor in eukaryotes. Instead, we have a series of multiple proteins called transcription factors. So let's go back down here. Here we have another diagram of a eukaryotic gene. All of this region here is promoter, and then all of this region here that's listed as gene A is what we're going to call the transcriptional unit. All right, what I hope you can see is that there is a whole slew of additional proteins, kind of like sigma needed to bind in our prokaryotic model. We have a whole bunch of different proteins that are binding here. And these proteins here that are binding allow RNA polymerase, which is this guy right here in this diagram, to recognize the beginning of the gene, okay? So these proteins that have started this process are called transcription factors, or your book calls them general transcription factors. Okay, again, what they do is they attach to the promoter of genes, and that allows RNA polymerase right here to recognize the beginning of the gene. Now, you can have different transcription factors for different genes. You have a whole slew of them. We've actually studied a transcription factor earlier in the semester. We just didn't call it that. I would like to draw your attention over here to this diagram that y'all have seen before. This is a diagram of steroid hormone signaling. And you might remember it as a lipid soluble hormone. Remember way back talking about cell signaling. Now let's walk through what's happening here, because what's actually happening is we are going to use a transcription factor to get this party started. If you remember, this is our steroid hormone here, and it's circulating in your bloodstream bound to a carrier protein. Because steroids are lipids, so they don't travel well in your blood, which is water. So they have to be escorted through the blood using a carrier protein. Once this carrier protein and steroid reaches the cell membrane, the steroid can diffuse right through this cell membrane, because remember, the steroid is a lipid and the inside of the cell membrane is a lipid. So diffusion through this membrane is easy peasy. Once the steroid arrives inside the cell, it attaches, see, this is a receptor here. You can see it's got one conformation before the steroid arrives. Once the steroid diffuses into the cell, it attaches to the receptor here, and that changes the conformation of that receptor. This steroid receptor complex then migrates to the nucleus, where it binds to the DNA of target genes to initiate transcription. Where is it binding? Well, y'all, where this thing is binding is in the promoter region of target genes. So, in other words, this receptor is a transcription factor, and this transcription factor recognizes promoter sequences for genes that respond to steroid hormones. So we've actually already introduced this concept. We just didn't know what it was called. So what this transcription factor here is doing, once it binds, it's sort of acting like a flag for RNA polymerase to come along and then make a copy of this gene. Maybe this is a gene involved in turning on glucose glycogen breakdown or something like that, okay? So that is one. So we already had the fact that RNA polymerases are different in eukaryotes, okay? So I'm going to put RNA pols differ. A second difference in eukaryotes is that they use transcription factors instead of sigma, because their promoters are way more complex. So they have a whole lot more proteins that recognize promoters than bacteria do, all right? And then the third thing that I have on here 4, why do I have it as 4? 3 RNA polymerases. Promoters are larger, more complex. Boop, boop, boop. Okay, promoters are larger. Okay. I don't know what 3 was supposed to be. I think I might have just misnumbered this. Okay, so 3. Another difference in eukaryotes is that at the end of the mRNA's transcript, remember how at the end of the transcript in eukaryotes, we had a hairpin. In prokaryotes, in bacteria, we had a termination sequence. And that termination sequence caused what we called a hairpin in our mRNA. So that looked something like this. And that hairpin actually caused the mRNA to dissociate from RNA polymerase, thus stopping the reaction. Eukaryotes stop transcription a little differently, okay? At the end of the mRNA's transcript, there is something called a polyadenylation signal, which I've already mentioned to you. And that polyadenylation signal is a signal to cut the mRNA. We're later going to use this polyadenylation signal as an enzyme binding site to add a whole bunch of letter A's at the end of the mRNA. So after transcription occurs, we're going to talk about this in a second, but this polyadenylation signal also acts as an enzyme binding site. And this enzyme's job is going to be to add what we call a poly-A tail to the end of the mRNA, okay? And then the last thing that's different in eukaryotes, and that would be number 4, since I've been misnumbering, is that transcription and translation are separated in time and space. I'm going to ask you to put a pin in that because we're going to come back to this when we get to the translation part of our lecture. All right, the last thing that I want to talk to you about in eukaryotes has to do with the fact that our mRNAs are copied from DNA that looks very different in eukaryotes than it does in prokaryotes. So let's remember that the structure of a eukaryotic gene has some untranslated regions, as well as the fact that we have exons, which are excellent, and so they code for protein. Introns are insignificant, so they are non-coding, code, non-coding, code. So in order for us to make a functional mRNA, we have to do some processing where we edit out the non-coding regions or introns. Okay? The way that we figured out this was happening is that we noticed that newly transcribed eukaryotic RNAs were not functional. And if we compared the eukaryotic RNAs in the nucleus, they were longer than the RNAs that are in the cytosol. And the reason for that is because we've edited out these regions here while this thing is still in the nucleus. So this process of editing out the introns is called splicing. And here's how it works. You can see here that we have an exon and it's got an intron in between, okay? What happens to get rid of this intron? Remember, we want to get rid of this. What happens is we have some small proteins that bind on either end of the intron. These proteins are actually a combination of protein and RNA. And so they are called small nuclear ribonuclear proteins, which is a terrible name, but we in molecular biology abbreviate this as SNRPs is how we say this. Oops, hold on. SNRPs, small nuclear ribonuclear proteins, abbreviated SNRPs. Now, what happens is these bind on either end, and then they form a loop here, as you can see, along with another enzyme. Together, this forms a feature called the spliceosome, which essentially works as a cutting factory to cut out this intron piece of transcript and glue together the exons. So here you can see that we have an edited mRNA, and this edited mRNA is a mature mRNA because now it only contains information that's good. It only contains coding information. Notice we still have a 5' and a 3' untranslated region, okay? I am going to ignore this right now. I'm going to save this for when you get to me in genetics because there's a lot of information in this chapter. So we'll talk about alternate splicing once you guys see me in our genetics course. All right. So we now have a situation where our mRNA transcript consists only of coding exons right here. So all of the introns have been eliminated. But we're not quite done manipulating this mRNA to make it a final product in eukaryotes. There are two other things that happen in eukaryotes to make a final mRNA product. The first is that at the 5' end, we add something called a methylated cap. That has to do with a methyl group, which is a CH3 chemical group. But basically what we have is a guanine along with a methyl group that gets added with some phosphates at the 5' end of the mRNA. Now, what does this do? Well, let's look down here at our notes. I tell you that, oh, okay, I don't have it in this notes. Okay, so what it does is it allows recognition of this mRNA by the ribosome for later use. Recognition. So this right here allows the ribosome later on to recognize this mRNA molecule. This also performs a little bit of a protective function. And I'm going to come back to that idea in just a second, okay? We also, as I mentioned earlier, have this poly-A signal right here that exists in this 3' untranslated region. This poly-A signal acts as an enzyme binding site. And what will happen to this mRNA transcript is an enzyme will come along, it'll bind here, and it'll just add a whole bunch of letter A's. These are adenines. So polyadenylation, adenine, A. This is what we call a poly-A tail or a polyadenine tail. Again, the function of the poly-A tail is also protection. So what are we protecting our mRNA from? Well, it turns out that we have a whole bunch of degrading enzymes, okay? These degrading enzymes that exist in your cell are called ribonucleases. And their whole job is to break down mRNA. And the reason you have so many of them is once you make mRNA, the idea is you make it, you turn it into protein, and then you get rid of it. Why do we do that? Well, we do that because the conditions that cells are in change quite rapidly. So you might go from a situation where you're being chased by a bear to a situation where you've gotten into a safe space. So you no longer need all of the genes turned on that were previously on because your situation has changed. So you want this mRNA to not stick around very long so that you can rapidly change the function of the cell. In order for that to happen, we degrade our RNA quickly because we don't want things sticking around that we don't need. But we also, it's a fine balance. We want to degrade that mRNA quickly, but we don't want to degrade it so quickly that we never actually get to make any protein out of it. So this mRNA cap here, this 5' cap, and this poly-A tail protect this region here that's going to be translated from the munching, degrading action of ribonucleases that attack it at either end, okay? The ribonucleases start at the end of the molecule and then munch their way inward. And so these features act like a buffer to prevent the ribonuclease enzymes from degrading this thing too rapidly, okay? Now, once we have a mature mRNA, so let me go back up here. This right here, this diagram that I've made very, very messy for y'all, represents a mature eukaryotic mRNA. This mRNA is ready to be translated into protein, okay? So that's our next step. We are now going to move away from this idea of transcription and move into this idea of translation. And what we need to translate mRNAs into protein, whether we're talking about a eukaryote like an animal or plant cell, or a prokaryote like a bacterial cell, we need three things in order to accomplish translation. we need an mRNA transcript. We're going to need a ribosome. Okay, and then the third thing we're going to need is an intermediate that goes between the mRNA and the ribosome. And you're going to find out that that is a molecule called tRNA. Okay, so I asked you here where proteins are made. They are made in the cytosol. Okay, so they're made in the cytosol if you are a prokaryote. They're also made in the cytosol if you are a eukaryote. Now, why did I branch this way? Well, because remember that for prokaryotes, the cytosol and the DNA material, they're all in the same place because there's no nucleus. So the ribosomes exist and protein synthesis exists in the same area as DNA, okay? So I'll put protein synth and DNA / RNA are in the same place. If you're a eukaryote, remember you have a nucleus. So the ribosomes or protein synthesis, they exist in the cytosol. So protein synthesis is in the cytosol, but DNA is transcribed into RNA in the nucleus, right? So we're going to have a little bit of a difference in how translation occurs just based on the fact that the geography of these two types of cells is a little bit different. So let's look at how protein synthesis differs in prokaryotes and eukaryotes. And as usual, we will start by looking at eukaryotes. All right, so if we're thinking about, looking at prokaryotes, I obviously have not had enough coffee this morning. Come on. Nope. Why is it not? There we go. All right, let's go back to black. I don't know why it's changing things on me. I want to go to black. There we go. All right, perfect. So here we have an image of the DNA molecule or the chromosome, if you will, in a, I don't know why this isn't working. There we go. In a bacterium. And I hope what you can see is that there's a whole bunch of little globs that are coming off of this DNA molecule or chromosome in this bacteria. What these are strands of mRNA that are being made. And as the mRNA is being made, what you'll find is that the ribosomes attach immediately. So here we have DNA. The DNA is being transcribed by RNA polymerase at multiple places along this DNA. We have an mRNA transcript that's being manufactured, and immediately as it's being manufactured, ribosomes are attaching to it. And these ribosomes are immediately translating this into protein. That is happening instantly. Okay. This is called a polyribosome situation. Makes sense. We have multiple ribosomes attached to a single mRNA molecule. And then out here, this part here is the protein as it's growing, okay? So you can see that the numbers indicate the order in which the ribosomes attached to the mRNA. So this one, they're moving this direction. This one has a longer protein attached to it because it started here and it started trucking down. So it's made more protein than this one attached here, and it's in the process of trucking down. And this one attached here, and it's in the process of trucking down. So you can see these are all making a protein based off of this single mRNA molecule, but this one's a little shorter because it just got started This one's a little longer because it's been going at it a little bit longer. And this one's a little longer because it's been going at it just a little bit longer. Now notice here we have the three things that we need in order for mRNA, or sorry, in order for translation to occur. You can see two of them actually, let me go back. You can see two of the three things that we need in order for translation to occur. We have an mRNA molecule, which is being transcribed off the DNA, and you have a ribosome. What you don't see is the third thing that we're going to talk about here in a second, which is called the tRNA. Okay, so this is how this would happen in a prokaryotic situation where everything is all together. Now let's contrast that with how this would happen in a eukaryotic situation where things are separate. So I'm actually going to go to your website for your textbook, and I'm actually in your study area, and I'm actually going to show you some of these little videos here. This first one I'm going to show you is called the RNA processing video. And what I want you to notice is where the mRNA is made and then how it leaves the nucleus. So let's take a look at this.
As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus.
Okay, so what I wanted you to get, oops, let me go back here. Actually, let me go forward because I kind of want to pause it. Go for me. Okay, this is where I want to pause. Nope, dang it.
Are added to the end of,
okay,
forming a tail.
To pause was over
the
here.
mRNA.
There we go. So basically what that was a quick review of the RNA processing that happens in the nucleus. And then you can see that this mRNA is leaving the nucleus. So all of that is to say that this idea here, where you can have your ribosomes and your mRNA all together in a prokaryote, they are separated in eukaryotes. Now, let's get back to this idea of we see that we have an mRNA molecule, we see that we have a ribosome. What we need is a way to go between the mRNA and the ribosome to make this protein. And that's what we're going to talk about next. How does mRNA get turned into amino acids? So far what you've seen is that you have a ribosome here, and a ribosome can be thought of as a little hamburger molecule, okay? The mRNA threads through this ribosome, and somehow this ribosome takes the message that's encoded on this mRNA via nucleotides. Somehow this ribosome takes this message and turns it into a series of amino acids, okay? But how does that work? How can the ribosome possibly take this message and turn it into a series of amino acids? Well, our buddy Francis Crick was involved in this idea as well. He did a lot of work thinking about how DNA gets turned into RNA and how RNA gets turned into protein. So the initial hypothesis that people suggested was they had these molecules of RNA that they understood. And if y'all remember, we figured out that RNA was broken into 3 nucleotide segments called codons, and each codon coded for, it was a code for an amino acid. So what people originally thought was that as this mRNA molecule got fed through a ribosome, what they thought would happen is that these amino acids would line up directly, that somehow these amino acids interacted, maybe through hydrogen bonds or something like that, directly with the mRNA, and that then an enzyme or the ribosome itself would come along and form a bond between these amino acids. And that made intuitive sense to people, that somehow these amino acids would just line up and that there would be some kind of bond formed. But our buddy Francis Crick, he was the first guy that said, no, this does not make any sense. That can't be the case. And why did he say that can't be the case? Hopefully y'all remember that amino acids have something called R groups. Each R group on an amino acid, different amino acids have different R groups, and they all have different chemical qualities, right? They all have different chemical qualities. Some of them are polar, some of them are non-polar, some of them are acidic, some of them are basic. Here's the problem with this hypothesis. It turns out that these nucleotides on the mRNA, those guys are polar, all of them. That's how they form hydrogen bonds between each other, okay? So in both DNA and RNA, nucleotides are always polar. What that means is if you had a non-polar amino acid, there's no chemical way that a non-polar amino acid could interact with this polar codon. So in other words, there was no way for this to happen chemically. So this hypothesis was wrong. What Francis Crick instead suggested was that there was some kind of an adapter, some sort of an intermediate molecule that on one end interacted with the mRNA, and on the other end, it interacted with the amino acids, okay? So what was this adapter molecule? It turns out to be something called tRNA, okay? So what we're gonna have is our ribosome, which is a hamburger with the big bun on top and the little bun on the bottom. We're going to have our mRNA that feeds through, and it's going to have a series of codons. And what we're going to find is that there's going to be an adapter molecule here. It's going to have an amino acid, an adapter molecule here that's going to have an amino acid, and then up there is where the amino acids will bond to form the growing protein or polypeptide chain. Okay, so let's take a look at these transfer RNAs. This is what a transfer RNA looks like. Now remember, I told you earlier that we had three different, in eukaryotes, three different RNA polymerases: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And remember I said that RNA polymerase 1 codes for ribosomal RNAs, or sorry, it copies ribosomal RNAs. RNA polymerase 2 copies messenger RNAs. And RNA polymerase 3 copies tRNAs or transfer RNAs. So RNA polymerase 3, what its job is to copy genes, DNA, that code for this particular type of RNA. Now, here's what I want you to notice about this RNA. It is a single strand of RNA that is able to fold upon itself, okay? So here is our single strand of RNA, but what it's able to do is to fold up on itself because there are regions that have complementary base pair sequences. So there are bases here that complement one another. There are bases here that complement one another. Here, here, okay? These form hairpin loops. We've already seen a hairpin earlier in our discussion. Now, sometimes students have a hard time visualizing how RNA can be a single molecule but end up with this weird structure. So once again, let's go back to your textbook site and let's look at RNA folding and how that can happen, okay? So here we go.
When the nucleic acid is relieved of the constraint of being sort of lockstep with another strand in a double helix, it's free to take on all kinds of shapes because it can interact with other parts of itself. So the structures that RNA can achieve have a wonderful variety, more on the order of what a polypeptide or protein molecule can achieve. And this is what allows RNA to be a biocatalyst rather than simply an informational molecule.
Okay. So I don't know if you understood what he was talking about there, but what I wanted you to notice, go back before this guy here. Oh, somewhere in there. Yes. I wanted you to notice these regions of complementarity. So this is a single strand of RNA, but because it's all by itself, it collapses onto itself and regions of base pairing emerge where you can create these hairpins. This is actually a tRNA, just a different representation of it than what you're seeing here. But this is a single RNA molecule that can collapse in on itself. Now, let's notice what's happening with this RNA molecule, okay? On one end, we have a series of base pairs. Let's go with a different color. We have a series of base pairs here that complement a codon. So here we have the codon ACU. This region here is the opposite of that codon. It is called an anticodon. So the anticodon is the complement, complement of the codon. All right. So at one end, that's what we have. At the other end, we have this sequence here, this ACC sequence. That is an enzyme binding site. And it allows attachment of an amino acid. For this amino acid, this sequence allows attachment of the amino acid serine. This anticodon is always paired with this amino acid serine. So you can envision how this is going to work. Here's our ribosome. It's two parts. Looks like a hamburger, okay? With a small bun on the bottom and a little bun on the top, we're going to feed through an mRNA molecule. Boop, boop, boop. Here's some codons. Here is a codon that is our ACU codon. We're going to end up with a tRNA molecule here. Oops, I'm doing a terrible job drawing this. Sorry for any artists in the room. Let me erase this guy here. Oh dear, that's not what I meant to do, but all right. We'll go back and redraw this. You guys get where I'm going, I hope. Okay, the anticodon here would be UGA, and at the other end would be a serine. You can imagine that if we move this over and we add another tRNA molecule here, okay, and let's just say we have a, I don't know, a phenylalanine here, then you can link these two things up to form a protein. So transfer RNA is the same thing as the adapter molecule that was mentioned by Francis Crick. It accomplishes that job, okay? So once you have a ribosome, an mRNA molecule, and a transfer RNA, you have the entire recipe that you need to initiate transcription of your mRNA into a protein up here, okay? So let's focus in on that process and how it happens, okay? We're gonna skip this slide, just is talking about how this is attached. Don't worry about that. All right, so let's focus in on translation. Let's focus in specifically on how ribosomes make proteins. Now I've been drawing for you a ribosome as a hamburger with the big bun on top and the little bun on the bottom. What is this hamburger bun made up of? Well, I hope you remember from earlier lectures that ribosomes are made up of both RNA molecules This RNA molecule that's in a ribosome is called rRNA. It's also made up of a series of proteins. So these are all the proteins. And what happens is these proteins and this ribosomal RNA, they all get together to make one giant macromolecule, okay? So in this example, we have two different RNA molecules, a little one and a big one, along with several proteins that are making our top bun. And then we have another RNA here, along with several proteins. They mix up to make our bottom bun. We have it again here. This is a eukaryotic set. This is a prokaryotic set. Notice some things. Both prokaryotic ribosomes and eukaryotic ribosomes are both consist of RNA plus proteins, RNA plus proteins, okay? Both of them have a large top bun and a small bottom bun. These different buns that I've been talking about are called subunits. So both prokaryotes and eukaryotes have large and small subunits, okay? What is different is that prokaryotic ribosomes have a little bit different proteins and different RNA than eukaryotic ribosomes do. Consequently, they are slightly smaller, but they function in exactly the same way, okay? Okay, so here we have, let me go back to my draw here. Here we have a blow up of a ribosome. So I showed you this with the large and the small subunits. And here you can see we have our large subunit here, our small subunit here. And the way that these ribosomes are arranged is so that the mRNA molecule threads between the top and bottom buns of our hamburgers, so between the large and the small subunit. You can also see there's some docking areas here for some tRNAs, and then we have a tunnel here where our new protein is going to emerge, okay? And this is just a more realistic rendition or rendering of what that would all look like up close. Now, let's go ahead and look a little more closely at this interaction between the mRNA in the ribosome and these tRNA molecules. So I want you to notice, here's our mRNA right here. And I mentioned we have 1, 2, 3 docking sites where we can have tRNA molecules occupy. One is called the E site, one is called the P site, and one is called, oops, I don't know why I wrote S, one is called the A site, okay? E stands for exit. P stands for peptidyl. Remember that the kind of bonds that we make when we make a protein are called peptide bonds. So that's where that name comes from. And then the last one stands for, the A stands for aminoacyl. Now, where does that come from? Well, it turns out that when you have a tRNA, here's our little tRNA right here. When you have a tRNA that has an amino acid attached to it, the formal name for that is an aminoacyl. Acyl RNA, okay? So these 3 sites basically are going to be where our tRNA reads the codons on our mRNA molecule, and the anticodon lines up. Therefore, our different amino acids are going to line up. Let's look at how this process of translation occurs, okay? So the first thing we're going to do is we're going to divide translation into 3 different stages, the same way we did transcription. And notice these stages have the same names. We're going to get the party started with initiation. We're then going to spend some time elongating the protein molecule or the peptide chain that we're forming. And then we're going to finish the party with termination. So let's start out with this process of initiation. What happens in initiation is that the mRNA molecule comes along and it's going to bind to the small subunit of a ribosome. Now, how does that happen? If you're a prokaryote, usually there is a sequence embedded in the mRNA that matches a region of the ribosome. Also, if you're a eukaryote, that guanine cap would also align down here. But either way, what eventually happens is that your mRNA here is able to attach to the small ribosome subunit here, okay? The next step is that we're going to have our first tRNA bind. And you remember that our start codon is AUG. So the first tRNA that's going to bind is going to have an anticodon right here that is complementary to our start AUG codon. So here's UAC, and here's our tRNA, and you can see that we have a methionine at the opposite end. Then we're going to close the lid on this ribosome. So we're actually going to add the large subunit on top, or you can think of that as putting the top bun on the hamburger bun. And when we do that, you'll see that we have a tRNA loaded in this middle space called the P space or the peptidyl space. So I hope you can imagine what's going to happen next. This is initiation. This is step one. To get elongation started, what's going to happen next is we're going to have the next tRNA diffuse into this ribosome, and you can see that this tRNA has an anticodon here that is complementary to the next codon in the sequence. So this guy comes in and docks in this A position. So here you see it docked in this A position. Now what we have is 2 amino acids at the top here that are lined up with one another. So we can form a bond between these two, a peptide bond between these two. So we form a peptide bond between the peptidyl site and the aminoacyl site. So that's how they get their names, okay? Then what we're going to do is this ribosome is going to move down the mRNA in that direction. So we're going to move over. That means that this guy is going to move over here and this guy is going to move over here. So that's what you see. This guy's here, this guy's here. Hopefully what you can see is that the next stage in elongation is going to be another tRNA is going to diffuse in. That tRNA will have an anticodon that is complementary to this codon right here. It'll also have an amino acid here. We'll form another peptide bond and we'll move forward again, just like a conveyor belt. Now what happens here is as we move forward, any of these used up tRNA molecules, see we've already taken off the amino acid. This guy is used up. He leaves out of this E site. Remember the E site is called the exit site. So this is step 2. This is how we elongate our mRNA chain. So let's look. Here we see the elongation, just like I said. The next guy comes in, he lines up here, amino acids get bonded here, and this guy leaves out of the exit, okay? So that is your elongation phase. That will continue until we get to phase 3, the termination phase. What happens in termination is that you reach a stop codon. Now, hopefully you remember from way back in the beginning when we talked about our codon chart that we had 3 stop codons. Here's 1, here's 2, and there's 3, okay? Notice that there's no amino acid associated with these stop codons. So what that means, let's go back down here. What that means, come on, there we go, is that, oops, right here. If you have a stop codon, you get a transfer RNA that has an empty spot up here. There's no amino acid. So there's nothing for a bond to form with here. And what that does is it stops this process of transcription in its tracks. That causes the entire ribosome to dissociate. So we're going to have our top bun peel off from our bottom bun, and that's going to set our mRNA free, okay? So that, in a nutshell, is how translation works. Let's watch a real quick animation so that you can visualize that a little more clearly. So I'm going to click on this from your website for your textbook. Here we go. Now let's watch it in action.
The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids.
Okay, so let's just summarize all of that with a couple of charts. This chart here, which is provided for you in your PowerPoint, is a summary of transcription and then translation in bacteria. So those are our prokaryotes, and eukaryotes. So that would be plants, animals, fungi, etc. Okay. So you notice that we have all these differences here in things like promoter structure, RNA processing we don't have in bacteria, but we have extensive processing in eukaryotes. And then it talks a little bit about translation, okay? The last thing I want to do is just summarize everything that we've gone over with one real quick figure that I think is very helpful from your textbook. Hang on. No, I don't want that. I want to get off there. There we go. So let's just start at the top here. And you can see that what we're doing, we're starting out with the DNA gene that we want to copy into RNA because we're going to make a protein. So RNA polymerase is going to transcribe the DNA gene that we're interested in. If we're talking about a eukaryote, then you end up with something called a pre-mRNA molecule that is not processed yet. So you can see that we have exons and introns. We don't have a cap and we don't have a poly A tail. So the next step, if you are a eukaryote, is that this RNA gets processed, a cap gets added, a tail gets added, and we cut out these introns. So we now have a mature mRNA. This mature mRNA is going to leave the nucleus through some nuclear pores. Here we have it. It's going to go to the ribosome. Remember, the ribosome has a large and a small subunit. They're kind of showing it upside down here, but essentially it works the same. We're going to thread our mRNA through this ribosome, and as we do so, tRNAs are going to line up, and we're going to have amino acids at the other end that will fuse to form a polypeptide chain. Once this polypeptide chain is made, then it's going to undergo some folding. It might turn into an alpha helix. It might turn into a beta sheet. So this is the primary structure of your protein. After it's formed, it will automatically go into a secondary structure, and then this will eventually knot up to form some kind of a tertiary structure, thus making an active protein for us, okay? These are called the major steps of what we call gene expression. And what we mean by gene expression is simply turning genes into protein. So expressing those genes as protein that will eventually manifest itself as phenotypes, okay? One last thing that I wanted to mention, I did have a question from one of my students today that I thought was really worth mentioning. I want to go back up to this codon chart. She was asking me if the things that we see in this chart are codons or anticodons. The things in this chart are codons. So this is the sequence as you would see it on the mRNA, okay? Not the anticodon that you would see on the tRNA. Okay, so this ends your Chapter 17 lecture set. Hopefully this is all helpful and you'll be ready for your quiz on Friday morning. I'll talk to you soon. Bye-bye.